Hello and welcome to the World of Plants Tissue Culture. My name is Silva and I look forward to a fantastic journey of exploration and learning with you all. This course is designed as a part of an Erasmus Plus Key Action 220 higher education project, namely BioTeach. This project aims to bridge the gap between universities and industry and facilitate the transition from university to industry by bringing together biotechnology students and industry representatives through this course. Over the next 15 weeks, we will be looking at the principles of plant tissue culture. This is a set of techniques used to maintain or grow plant cells, tissues, or organs under sterile conditions and a cultural medium of non-composition. It is often used to produce clones of a plant in a method known as micropropagation. In the coming weeks, we will cover a number of once again. In the coming weeks, we will cover a number of exciting topics, including application of tissue culture in agriculture, obtaining virus-free plants, conservation and cryopreservation of plants, haploids, protoplast fusion, production of secondary metabolites, regeneration of transgenic plants, and much more. So, welcome to the world of micropropagation. Together we will explore the fascinating process of plant tissue culture, an important skill for budding researchers. I'm really looking forward to diving into this topic with you and hope you find it as fascinating as I do. Micropropagation plays a central role in the mass production of plants with desirable traits. It's like assembling a team of botanical clones, each with unique abilities and ready for scientific exploration and agricultural challenges. Micropropagation is therefore a cornerstone of modern agriculture, as it enables the efficient and rapid propagation of selected plant tissue. This technique plays a crucial role in maintenance and propagation of plants with desirable traits and ultimately contributes to the improvement of crops and biodiversity. Before we get started, we should familiarize ourselves with the most important tools: culture vessels, growth media, and a sterile environment. For example, imagine setting up a plant tissue lab. Our journey begins with the disinfection of equipment and culture media. Maintaining a sterile environment is essential for successful micropropagation. How do we select the starting material? First of all, we should find a robust plant specimen. It is extremely important because only healthy parents lead to robust offspring and thus create the conditions for success. And how do we recognize plant material that is suitable for establishing tissue culture? Well, it's not that simple. Somehow you have to be skillful to recognize the characteristic of a healthy and disease-free plant that is suitable for starting in vitro propagation. In the next phase, the selected plant material undergoes certain transformation. First, the collected plant material should be carefully cut into small parts, so-called explants. Those explants serve as the basis for the growth of new plants in order to cultivate a living and dynamic plant community. Now we have selected the suitable parent plants, but how do we prepare this raw material? Before cutting, we need to carry out a crucial step: surface disinfection. This precautionary measure minimizes the risk of contamination and ensures a clean starting point. We then place the explants on a nutrients-rich growth medium. This environment provides the necessary nutrients for the development of the new plants. To produce a medium for plant tissue cultures, the basic components such as mineral salts, basal medium, sugar as an energy source, and vitamins are combined in appropriate concentrations. Agar or agarose is then added as a gelling agent. The pH value of the medium is adjusted for optimal growth conditions. A pH value between 5.5 and 6.5 is most suitable, as the availability of nutrients is optimal in this range. Preparing media in the microwave is a quick and effective method of dissolving the ingredients. To do this, we place the mixing container in the microwave and heat it at intervals, stirring occasionally until all the ingredients are completely dissolved. After heating, we sterilize the medium, usually by autoclaving, to ensure that all potential contaminants are completely destroyed. The next step is to enrich the growth medium with specific plant hormones. Plant hormones stimulate the growth of the explants by promoting the formation of their shoots and roots. Of course, we should provide a controlled environment with optimum temperature, light, and humidity in growth chamber. This ensures ideal conditions for the growth and development of our plants. Once again, of course, we should provide a controlled environment with optimum temperature, light, and humidity in the growth chamber. This ensures ideal conditions for the growth and development of our plants. Regular subcultivation is necessary during the development of new shoots. This involves moving the plants to a fresh, nutrient-rich medium to promote their growth and development. Healthy roots are essential for transplanting. We should promote root development. One way to do this is to adjust to hormone concentration in the growth medium. Once again, since healthy roots are essential for transplanting, we should promote root development. One way to do this is to adjust the hormone concentration in the growth. Once again, since healthy roots are essential for transplanting, we should promote root development. One way to do this is to adjust the hormone concentration in the growth medium. As soon as the roots are well established, we wash them well under. Once again, as soon as the roots are well established, we wash them well under. Once again, as soon as the roots are well established, we wash them well under running water and carefully plant the macropropagated plants in a soil substrate. The next crucial step is acclimatization. During this process, we allow the plants to gradually acclimatize to their new environment outside the vessel. This step strengthens the plants for successful growth. Internal endophytes are certain bacteria, mycoplasmas, nematodes. Once again, internal endophytes are certain bacteria, mycoplasmas, nematodes, viruses, and fungi. They live inside plants and colonize them without having any harmful effects on the host organisms. However, their presence in the tissue of explants can have a negative effect on in vitro propagation. These internal colonization appear as a cloudy white or colored surface on the growth medium, or in some cases, once again, this internal colonization appear as a cloudy white or colored surface on the growth medium, or in some cases, unfortunately, may remain invisible. How do we help plants in the fight against harmful bacteria? Immediate action is crucial. Antibiotics are often added to the culture medium to overcome the challenge of bacterial infection in tissue cultures. When we have found a suitable solution such as antibiotics, we can celebrate victory over internal bacterial infections. This proves the effectiveness of maintaining healthy plant tissue culture. Once again, when we have found a suitable solution such as antibiotics, we can celebrate victory over internal bacterial infections. This proves the effectiveness of maintaining healthy plant tissue cultures. At the end of our journey, we can summarize that micropropagation is a revolutionary method for cultivating plant species. It allows us to propagate rare or endangered plants, increase agricultural productivity, and contribute to the preservation of biodiversity. Once again, at the end of our journey, we can summarize that micropropagation is a revolutionary method for cultivating plant species. It allows us to propagate rare or endangered plants, increase agricultural productivity, and contribute to the preservation of biodiversity. Congratulations! Once again, congratulations! You have experienced the journey from plant tissue selection to successful micropropagation. I would like to thank each of you for participating in this course. You're available. Once again, I would like to thank each of you for participating in this course. Your valuable contribution and commitment to scholarly research will not go unnoticed. Once again, I would like to thank you once again. I would like to thank you, each of you, for participating in this course. Your valuable contributions and commitment to scholarly research will not go unnoticed. Thank you for your efforts. Now it's your turn. We invite you to think about and explore the various challenges associated with tissue culture. In addition to internal infection, other problems can occur during micropropagation, such as vitrification or browning of the tips. What can we do if something like this occurs?